So I guess without further ado, I think we will start the second half of our workshop, where we will have first Kellyanne presenting about doing some analyses with chime, and hopefully walking through some other steps. And let her take over with that.
Awesome. Good morning, everyone. My name is Kellyanne. I'll be leading you through this workshop, if I can find my, there we go. So if you have any questions at any point, please, it might be easier to just call out my name because I'm probably not going to be looking up, but if you have a question, somebody else is definitely having that same question, so please speak up. But real quick, some background. So we're analyzing 16S data using chime. Chime stands for Quantitative Insights into Microbial Ecology. So what's really great about chime is it goes beyond just characterizing who is there. We're looking into ecologically important characteristics that, like Chris was describing, alpha diversity, beta diversity. What I like it for is for the quality filtering and for just quick exploratory analysis. When Damien comes up, you'll be seeing that we do a lot of stuff in PhyloSeq using R, and when you're doing publication figures, you tend to use R packages, but chime is really great for just quick, how does my data look kind of analysis. Also, just so you know, we'll be using chime 1 today, but chime 2 will be succeeding. Chime 1 starting January 1st, so chime 1 won't be covered anymore. But you'll see that a lot of the concepts will be exactly the same. The script layout will be pretty similar. You'll just have to learn a couple new tweaks. So, if you want to look at all the potential scripts that you can use, you just go to this web page, just type in chime, and it lists all the scripts. And then each script, so here I just took a screenshot of just one of the random scripts that we'll actually be using. I don't know if you can see very well, but at the top it says beta diversity through plots, so that's the script. Under description it says what we're going to be doing with it. And so if you want to do some type of analysis, you'll look through the scripts and go, okay, well, this is what I want. And so here's an example at the very bottom. Can anyone actually see this? No? All right. So we'll just, but we can still use this figure, and I'll go through it a little bit later. But at the top, it says your input arguments, and you have three required files for this script.
Excuse me, Kellyanne, we need you to talk into the microphone so we can catch it on the video.
Sorry. Okay, so the first required file is called an OTU table. This is really the most important file you're going to be using, and we'll go into what that is in a second. And then the second one is your mapping file that has all your metadata. So that tells all of your analysis, like, hey, this is what the sample is, and we'll go into that in a minute. And then all of these are just different flags, that ways that you can change your data. Depending on your question, like ways that you can change the script. So let's talk about real quick what an OTU table is. So Chris mentioned that an OTU is an operational taxonomic unit. This is just a way of saying a specific bacterium, because we're not always talking about species, sometimes we're talking about classes. So it's just easier to say OTU. When we have your OTU table, you have your columns are samples. And then on your rows are the different OTUs. And so your OTU table is just the number of reads of a specific OTU found in a particular sample. And then we have our mapping file. This is something that you generate. Using chime, sorry, I heard my name. Okay.
Right. I'm a professor, so I'll tell you, slow down. Okay, it's your speech. But like this OTU file, what is it? Is it an Excel file? Is it a text file? Where does it live?
So it's a biome folder. We're actually going to be making it. It's a special format because it has a lot of data in it. I kind of oversimplified it when I was showing like this. You can have it shown as a TXT file, but when we use it, it's this biome format. I forget what biome stands for, but you could Google it. But by the end of halfway through the tutorial, we'll have our own OTU table, and we can look at certain characteristics of it,
okay.
And then we'll analyze it.
So you have to construct this yourself?
Exactly, from your sequencing reads. So you know how Chris was saying, you get all your sequencing reads, and you do your quality filtering, and then you assign them to OTUs?
Yeah,
This is the table that you get that tells it what reads belong to which OTUs and like who those OTUs are. This only has numbers for OTUs, but it'll say like kingdom, class, order, like the actual taxonomic categorization. Did that make any sense?
no?
No. Okay, so your OTU table has all of the number.
I get what the end product is. I mean, that's obvious, but I have no, I don't appreciate yet, okay, and that you do sequencing, and then you've got this massive sequencing file, and you're going to stuff it into some program that spits out an OTU table?
Right, and we'll actually be going through that. I think it'll be easier once we actually go through the steps. But it's taking the reads and finding out how many reads belong to which specific OTU.
Yeah.
And so then you can do your preceding analysis.
Yeah, I think that'll be exactly what it is that once you get your reads, you will have, you're going to go through a set of processing steps. And then you end up with this. Then you'll get to the, table at this point. And she will go through that.
Okay, so back to the mapping file, there's just a few things that are necessary. These 4 categories that I've highlighted, sample ID, barcode sequence, linker primer sequence, and description, are necessary for your mapping file just for it to work. And it has to be in this order, so the sample ID, barcode sequence, and linker primer sequence need to be first, description needs to be last. It doesn't really matter for barcode. So barcode sequence and linker primer sequence, you'll see that there's just a bunch of X's. And that's because I don't actually have information for those, because I use the URI Sequencing and Genomics Center for my data, as probably most of you will, and they already provide me my data as demultiplexed, which means that my samples that were pulled together for the sequencing run are already separated into their respective files or samples. And so if I were demultiplexing, I would have information there. I don't, so I put the X's there. You still have to have them because there are points in your analysis where you want to merge or separate mapping files, and it's just have these files. It just makes your life easier. Notice also at sample ID that there's that little pound sign. He has to be there. It's just part of the process. I already explained the barcode sequence. Okay, and then you'll see there's this input file name, which is redundant. It's the same thing that's in the sample ID, but this is going to be important when we're adding labels to our sequencing reads, which will make more sense later. So just know that I have this column called input file name, and it has the same identification as the sample ID. Also, if you're ever wondering if your mapping file is correct, you can use the script validate mapping file. We're not going to be using it today, but it will check to make sure that your mapping file is correct, and it will actually spit out the mapping file that it thinks it's correct. I don't use it. I think it's a little oversensitive, but if you're learning, it's probably a good tool. So what we're going to be doing in this tutorial is, first we're going to trim our reads, so getting rid of low-quality base pairs. Then we're going to join the forward and reverse reads, and then we're just going to convert that file. We're going to do that for each sample, and there's 8 samples. And then once we have all of that done, we're going to take all of those samples, add those chime labels, and then do the OTU picking. And as I said earlier, I don't demultiplex my data because it's already demultiplexed. If you use Janet at URI, yours will also be demultiplexed, but if you ever want to, you can use this chime script demultiplex fastq.py. So that's it for that. Let's go into the tutorial, but real quick, I just wanted to bring this back up because this is actually the same data that we'll be using. So I just picked 8 samples. 4 of them were from a wild-type mouse, 4 of them were from a knockout. They're all fecal samples. And so that's what we're going to be playing with today. So while I pull this up, does anybody have any other questions? Okay.
So I have one comment, just for people, if you don't know the concept of demultiplexing, it's just that when you sequence nowadays, what you can do is you can sequence multiple samples together in one lane, and so each of those, each of those samples, the sequences from each of those samples have a specific barcode attached to them, so that once, the sequencer doesn't know, right? It just kind of reads through the sequence and it just comes out with these reads. And then once you have that, then you can go in and say, oh, my read has this set of sequences in the beginning, therefore it should be from this particular sample. So a lot of sequencing facilities will now automatically do that. They'll separate it out and give it out to you. But occasionally you will get a place where they're just going to give it all to you, and then you'll be like, I sent in 10 samples. Why do I just have 2 FASTQs or 1 FASTQ? And basically it means that they may not have demultiplexed. The other issue that may arise is that sometimes when you have these FASTQs, you may say, I sent in 10 samples, I got 10 files back, but I have this one giant file and really 9 other small files, which essentially means they didn't do the demultiplexing correctly. Or you may have not given them the right set of barcodes. That happens a lot too, where, or in the very worst case scenario, you may have put in the wrong barcodes and they're unable to do anything with the data anymore. So those are that's sort of the kind of background of demultiplexing.
Thanks. All right, so if you guys want to go into the Chime notebook, IPYNB. Was everybody able to find it? You should see something that looks exactly like this. Wonderful. So if you want to execute, just press Shift-Enter. Things like this first one didn't have any script, so nothing happened. But you might see if you catch it quick enough, before the number comes, there's a little star. That means the script is running, so just leave it alone if that's what's happening. And just go ahead and run this from IPython display script. We need that in order to look at some of the visualizations later. Okay, so now that that's started, we're just going to change the directory to go into the Kellyanne directory. And then you should get this, so it will tell you that you're actually in the Kellyanne directory. And we're doing that because that's where all the read files are. So look at what's in there. You can just do ls, and then you'll see these files. So there's this just in case, don't worry about that. That's something in case something goes wrong. We have our samples, so KD 1 through 8. We have our mapping file that I've already made. This next TerraPE thing.
Sorry, so why are you at like 9 versus like I'm at like. Oh, sorry. Why are you at like 9 at the bottom? Like, does that even matter? Like the numbers in the brackets?
It doesn't matter, but it is. So normally when I do this, it actually goes in order. I don't know why it's not going in order, but it doesn't matter.
Okay, it doesn't matter.
But very astute observation.
Well, I was just curious.
Okay, so we have that mapping file. That next Terra folder, I'll talk to you about that in a second. It's kind of redundant, pointless to bring it up now. But let's go into the first folder. So we're changing the directory into KD1. And then we're going to look inside with the ls. And then you'll see that there's 2 files. KD1, blah, 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 R1, fastq.gz. KD1, blah, 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 R2 dot whatever dot .gz. So the R1 is your forward read, and the R2 is your reverse read. That's just how sequencing centers do it and how the software recognizes which read is which. So then first thing we're going to do is we're going to trim the reads. This is going to generate 4 output files. So let's look at the script real quick. So you'll see that I put in the program trim-o-matic. I put in PE, which stands for paired end. And then there's Fred33. All of these things that I'm talking about, putting in the PE, putting in the Fred, all of these inputs that I'm talking about, you can see all of this on the trim-o-matic website, which will spell out like what you're actually going to put in. And so first, after Fred33, we put in our forward read, we put in our reverse read. And then we have our 4 output folders: output forward paired, output forward unpaired, output reverse paired, output reverse unpaired. And so this is going to spit out, after the trimming, which of the forward reads were able to survive with their reverse read so that we can eventually pair them, and which ones weren't. Then we have our Illumina clip, which is not necessary because we already have demultiplex data, but I just threw in the next Terra folder just in case something happened. What's really important is this head crop, and that's taking off I have it set so that you're taking off 18 of the first bases. And the reason why I do this is because even though our data is demultiplexed, when you use your PCR adapters to amplify your 16S region, you're recognizing a conserved region. And the problem with that is when you're doing your binning and you're assigning to 97% similarity, but you have this known conserved region, you're biasing your binning. And so this will get rid of that conserved region so that when you're saying 97% similarity to a certain OTU reference, then it's an actually true representation. Leading and trailing. Yeah. You have to get the box from Jackie. Sorry.
It's just a note for you. I don't think anyone here knows exactly what kind of data you're looking at.
Oh, that's smart. Yeah. So
And that's why none of the, like the trimming or the barcoding or the 16S stuff may or may not be making sense to everyone. And please talk slower.
you know that I can't.
You can do this.
Like the one person in this room who knows.
Oh.
So
Kellyanne, I have a question.
Yeah.
So the trimming is basically just to get rid of the parts that are the barcode, or is it getting rid of the parts so that you can align the two reads?
it's getting rid of the low quality bases. It also gets rid of the barcodes if you want.
But then how does it make the call what the low quality bases are?
So all of that is, I'm about to tell you.
Okay.
That's, yeah, like it's kind of a long script, I'm sorry, it's coming. So let me just touch on Nimrata's thing. So this is 16S sequencing data, and as Ashok was saying, I amplified the variable 4, variable 5 regions. What else did you want me to say?
So you have 16S data. Each one of those samples is KD1, whatever, 1 through 8
Each
or
sample
whatever
is, yeah,
it is.
KD1, KD2, KD3, those are all samples.
And right now, you're just taking that one community file.
Right.
Each sequence was barcoded. The sequence read contains the barcode.
Sequence
Still,
read does not contain the barcode. The
It does not
barcode
contain-
was removed in demultiplexing.
Okay,
The
so
Headcroft thing was just taking off the region that you added your PCR adapter to, just a conserved region, because we don't want conserved regions.
so when you're amplifying, you're starting from a conserved region, and the middle part is the variable region that gives you the actual like taxonomic classification
Exactly.
information. So you're removing the conserved region from both the forward and the reversed,
Correct.
along with also trimming off low quality reads on
Exactly.
either end.
But the conserved region, is that from the barcode, or is that?
It's it's not really a barcode
Oh,
per se.
there's a part that's always conserved, and then you're looking
When
at
you're
the
adding on your
variable
first
region.
PCR, yeah,
Okay,
exactly.
yeah. Otherwise, you wouldn't be able to PCR it.
Yeah. Exactly, that's exactly it. Okay, perfect. So this is really, this is why I use Trim-O-Matic as this next point, which is the sliding window, because you could do this in Chime. I think Trim-O-Matic is a little bit more biologically relevant, and that's because you have this sliding window. And so it's actually going to read every read you have, and it's going to make a window based on what you say. So this 5 means it's going to read every 5 base pairs and take an average of the Fred score. And I'll tell you what the Fred score is in a second, but you can change this to anything you want. So Fred score is just the log of the error probability, and so what you just need to know from that is a higher Fred score means there's more of a chance that that base was sequenced correctly. So I have it set so that if it reads 5 bases and the average falls at 18 or lower, it's going to trim that read. I also have it set for a minimum length of 100. And so these are mostly variables that you can play with. If you read the Trim-O-Matic paper, you can read more about it. But we're just going to run it real quick. So press Shift, Enter, and then all of this stuff will come up. You'll see the stars there, so it's still running. This is all stuff about the adapter that we don't really care about. What we really care about here is, so both surviving, so about 83% of my reads, my forward and reverse reads survived. Looks like I have good data. That's exciting.
Wait,
Not that-
Kellyanne, what do you? Okay, how do you get there? The shift enter.
Yeah, so Shift Enter is going to run any of these scripts.
When you're clicked on that thing.
Yeah, and so if you click on the first one and only press Shift Enter, you don't have to keep on clicking to new ones. It will just automatically move to the next one.
Hey, Kellyanne, quick question. What cutoff of both surviving is unacceptable?
Sorry, what was unacceptable?
Yeah.
I mean, that's up
Like,
to you.
what's your minimum percentage you're aiming for no matter what?
I don't have a minimum that I'm aiming for. I don't really ever fall below 80% for my stuff.
Okay.
I think it really depends on your question. I think with my type of analysis, I'm more worried about characterizing the whole community rather than getting to specific genuses. So I'm a little less stringent, but I'm able to maintain more reads. But definitely if you're trying to be like, this is exactly who is here, I want to get this to the family level, then you want to change these to make them more specific. Okay, so we have probably successfully trimmed, and it should say trim-o-matic PE completed successfully. Beautiful. We can actually see how this quality changed by doing a FASTQ. So first, run this next script, FASTQ, KD1, blah, blah, blah. You'll see that it finishes. And then if you run the iframe, immediately under it, you'll see this FASTQ report. And then all I use this for is going down to this per base sequence quality. And so this was the read before we trimmed it. And you can see the x-axis is the length of the read. So towards the end of the read, the median falls. This is a forward read, so it's not the worst thing I've ever seen. But if you get out of that and run the next FASTQ on the output forward paired, we'll see what the forward file looks like after trimming. If you then run the iframe immediately below that. And so you can compare, and so looking at my per base sequence quality, it looks a lot better. It's been trimmed. I'm very happy with that. We can do the same thing with the reverse reads. Reverse reads are notoriously worse, and so don't have a heart attack when you see this file, which is really bad.
Do you know why the reverse reads are consistently worse?
No, Damien, do you know? I've never known.
It's because of, that's how Illumina, I think because of Illumina sequencing itself is that, that's, I think as the read flips over, I think it's that the polymerase doesn't do as well as longer as it gets to the end of the read, but it's more a sequencing technology thing. So if you have 150 paired end reads, usually you see that the second read is worse than if you have, say, 50 base paired end reads. But I think it's also depends a lot more on the size of your fragment that goes into the sequencer as well.
Cool, so that was the reverse read before we trimmed it, and then if we FASTQ see the output reverse paired, and then iframe, which will visualize it, we'll see it looks a lot better. And you can do that for all your files, if you so choose. It's a really good idea to do that when you're trying to mix up your, or try to figure out what kind of quality filtering you want to do. So let's go inside and look at what's in our folder. We're still in KD1, and you can see there's a lot of files that are just dirty. So I'm just going to ask you to remove them by running the remove HTML and remove the zip. So that, I don't know why that keeps happening. So that when we look inside KD1, we see the two original files, the forward read, the reverse read, and our 4 output files. So now we want to join the forward and the reverse read. And so we're going to do that with this join paired ends. So why don't you go ahead and run it with Shift-Enter while I explain it. So like I said, we're joining the forward trimmed read and the reverse trimmed read. And then there's a couple flags that you can add. So it requires you to have an output flag, so the -O, that's the output, so it's going to make an output called paired. The J flag is the minimum overlap of base pairs. I've set it to 30. This seems to be pretty standard. Same for the P flag, which is the maximum difference in base pairs allowed, so I put 10%. All of these and other flags that you might want to modify are on that chime scripts webpage that I explained earlier. And so that should be done. So if you do ls, you'll see that you now have an output file called paired. And we can look inside.
Kellyanne?
Yeah.
So it has this whole jumble of reads that are now trimmed, and every read has a complement, an exact complement somewhere.
If you're looking at the output forward paired and the output forward reverse. those guys have their pairs in the opposite file. And Trimemetic knows which forward goes with which reverse.
So this is a really dumb question.
I love
So in the same sequencing run,
Mm-hmm.
you are sequencing with ones with forward and ones with the reverse primer, or are these two runs? for
No,
every
it's on the same run, and when we go back to lab, I'll show you, there's a really great Illumina video where it shows, so first you sequence, sorry, I actually see the video in my head. So you sequence the first strand, and then the reverse one falls off, and it falls next to it, and then it knows because of proximity based on the clustering.
Okay.
So, I mean, the way Illumina sequencing works is that, every read pair is usually one read is on the forward strand and the other read is on the reverse strand because of the way the bridge amplification itself works. So you always get one read for a given read pair, one read on the forward and one read on the reverse strand.
So it has they're next to each other, so it knows who's paired with who.
They're not next to each other. You can think of it as like, if you think of sequencing a fragment, right? It's just one fragment, and you're sequencing the two ends of the fragment. So the first end will be on the forward strand, and the second end will be on the reverse strand of that given fragment. Does that make sense? So it'll be like the complementary strand to those base pairs at the end of the fragment.
So it's not searching for the reverse complement. It knows which.
It knows which. It's not searching for a reverse complement or anything. All it's finding out is that which reads are coming from the forward strand and which reads are coming from the reverse. It's not trying to do a reverse complement or anything like that.
Okay, so looking inside the paired file with the ls paired, we can see there's 3 files. So the fastq join.join.fastq, that's our joined sequencing reads. Un1 and Un2 are the ones that weren't able to be joined. And so we're going to take that paired file, that fastq join.join is going to be the file that we use next. So now we've trimmed and we've paired our forward and reverse reads. And so now we're just going to convert the file type, which isn't a big deal, but I thought this would be a good chance to look at the data. So what we're using right now they're called FASTQ files, which is a FASTA file plus a quality file. If you run the head paired FASTQ join FASTQ script, you can see that this is what you're looking at. So this is one. There we go. So this is one sequence, right? But at the top part, this is our quality. So these are the letter representations of the Phred score. So this will tell you the quality of each base. This is some identification information, and then this is your actual sequencing read. When we convert the file, we're just going to be separating the quality from the actual sequencing. So go ahead and run the convert fasta qual fastq.py file, and that's going to take a second. So let me explain what that is doing real quick. So I told it to run the script, convert fasta qual fastq.py. It asked me for my forward strand, so because I'm not in the paired directory, I had to indicate, look inside the paired directory and grab this file, the fastq join.join.fastq. And then I had to tell it what way to convert it, so fastq to fastaqual. You can also use the same script to combine fastaqual and make it into a fastq folder. That's obviously not what we're doing, but these are things that you can do. If you look at the chime scripts page, it describes all of this. So that is currently running. That might take another second. Are there any other questions?
Kellyanne,
No,
why do you want to pair them?
why do I want to pair them? So some people, and I found out Rob Knight does this, only use a single reads. And so in theory, you could just sequence the forward strand and you can identify your OTUs that way. However, I like the idea of having to have the paired as kind of a safe net. Like I trust more, there's less likely sequencing error to influence the changing of your OTU identification if you're requiring a paired read versus just reading like a single read. But some people are doing single. I think paired is better, but who am I to tell Rob Knight what to do? Okay, so that should be done. So if you want to go ahead and press LS, that's going to show you your 2 new files. So you now have fastqjoin.join.fna and .qual.
How did you know it was done?
So this little star went away.
Oh, okay.
So you should now have a number next to the front of the script.
I feel like I queued up a couple lines right now without my stars next to them, but it's still, you get to number.
So it might still be running for some of you guys, but for those of you that it's done, you can go ahead and do the head. That's just going to show you the beginning of the script. So we're not looking at, not the script, sorry, the file. So we're not looking at the whole file. And so now you can see that we just have the sequencing reads, the base pairs. We no longer have the quality scores. If you wanted to look at that, you can go into the quality file. Okay, so now we have done the 3 things that we wanted to do for the first file. We trimmed, we joined the paired, and we converted the file. So now we need to get out of KD1 by changing the directory with the 2 dots and the slash. That's going to bring you to the previous directory, or like 1 step up. So that's going to take you back into the Kellyanne directory. And what I'm going to ask you guys to do for the next 10 minutes or so is to run all of step 4, which is going to take you through each of the files, KD2 all the way to KD8. It's going to trim, it's going to join, and it's going to convert. And just go ahead and do that. But the thing is, make sure that you understand what's being inputted, where you are, because this isn't going to be exactly the same when you do your own stuff. So it's important to know what you're putting in. Anyone still running? Okay. The good news is if you didn't finish running, there's something we can do. So starting with step 5, we're taking all of those joined files that we just made. We had just converted the files, and what we're going to do is, well, first we're just going to make a directory, just making a folder. It's going to be called seeks. And then we're going to go inside each of these folders, so KD1, and then, for an example, KD1, and we're taking that fastqjoin.join.fna file, and we're going to move it into our newly made seeks file. And at the same time, we're going to rename it KD1. And the reason why we're doing this is because right now, as that file is in KD1, it's called fastqjoin.join.fna, right? Well, in all of these files, it's called the same thing. So we need to add some kind of unique identifier. And the unique identifier needs to correlate with the sample ID and input file name. Yeah, input file name that you guys might remember from that mapping file that I had earlier. So that's what this is going to do. So go ahead and run that. It makes the directory, and then it goes inside each of those files, takes out that file, puts it in a new one, and changes the name. And so if you want to remember what I was talking about, you can print the mapping file with the cat mapping file function. Oh, wait, I have to stop that. There we go. And so, sorry, the spacing is a little off, but your input file name, which corresponds with your sample ID. And then, if everything worked, you should be able to look inside your seeks folder with the ls function. You're not going to see it on mine because it didn't work on mine, but it will show you the different sample names, KD1, KD2, KD3, all the way to 8. And if everyone's ready, we can add the chime labels. Okay, so the chime labels are those unique identifiers from that input file name that we were just talking about. And so every file now has its input name as the file name. So we're just going to run this add chime label script. You put in the mapping file, you put in the directory that has all your samples, and you told it which column to look at as to which label to put on which read. This will probably make more sense once the file is done and we can actually look at, actually, hold on. Okay, so I won't be able to show you, but so once the script is done, you'll run the head combined seeks.fna file. And so you'll see that before I showed you, you just had your sequencing reads, you just had your bases. But this time, it will have your chime label, so it'll say KD, whatever, 1, 2, 3, 4, 5, 6, 7, 8, whichever sample it came from, and then it will have your sequencing reads. And this is important because after the script, you're going to have one large combined file. That's the combined seq.fna. So those are all your sequencing reads for all 8 of those samples. And they're all mixed together, so you need each read to have its unique sample identifier. And so if you're done with that, go ahead and go to step 7 where it says closed reference OTU picking and run that script, the pick closed reference OTU, because it takes about 10 minutes if everything worked. Is everybody at the closed reference OTU picking part? No? Where are you, Jackie? No? Just not even? That's fair. No, okay. Is anyone else other than Greg getting an error? Okay, so hopefully everyone is picking their closed reference OTU script, so this is a great opportunity to talk about what it actually is. With my data, it's from the mouse fecal microbiome, so it's very well characterized. And so what the script is doing is it's looking at a chosen reference database. The default for chime is green genes, so it's looking at the green genes database, and it's looking at all my sequencing reads, and it's trying to map it to a sequencing read on this database with 97% similarity, and that is how my reads get identified as a specific OTU. And so all we needed for this was the input, which is the combined seqs file that was generated from adding the chime labels, and then just putting it as some output name, so I called it closed ref. Hey, Ethan.
What does
You have the box literally right next to you.
What does the program do with unmapped reads or reads that aren't in your reference genome?
They automatically get tossed. So I don't know too much about it, so I'm not going to go too much into it, but there are other things you can do. So I do closed reference. There's also open, and there's also de novo. Damien's going to talk about de novo? Yeah, okay. So because my model system's microbiome is well characterized, this is perfect. It's great for other people that aren't so lucky. I have a friend who's studying like the barnacle microbiome, and so she has to do like, I don't even know what she does.
I'm a little confused about the mapping file.
The mapping file, do you want to talk about it after you get your box? Sorry, I saw Trisha and I was like, ah.
So I'm confused. So we did the sequence directory where we got all the FNA files on and labeled them based on the sample that they corresponded to.
Right.
Then we did a print mapping file. Did it create that mapping file?
No. You created the mapping file
Way before.
Like when you
Okay.
were setting up your sequencing run, you were like, okay, in sample 1, it's going to be this. This is my metadata. I just had you print it because I wanted people to look at the column name that I was referring to.
So when you said you added a chime label, you had, you said, look at the mapping file, and so its source was the mapping file, and the thing it was modifying was the sequencing something. No, so what is a chime label, essentially?
The chime label is just putting on a unique identifier onto each sequencing read to tell it which sample it came from. So that's
Okay.
why each sample is, why each chime label is KD1, KD2, because those are the sample IDs.
Okay.
And so because you have all your files separated right now, and then when you add the chime labels, it combines them. And so you need to know which read came from which sample. And by you, I mean the software.
Okay, yeah, that makes sense. Thank you.
Anytime. Okay, so mine isn't working, so I will need someone else to tell me when the closed reference script finishes. Is anyone actually able to get the closed reference script working? Okay, I got a couple nods. This is promising. Oh, I even got a thumbs up. Thanks, dude. So hopefully people are finished, and for the sake of time, we'll we'll get going. If you want to look inside your new folder that you just made called close ref, it should do something. Oh, okay, so yours will actually say, mine didn't work, but yours will say something OTU table.biome, so that's your new OTU table, congratulations. It'll also say 97-OTU.tree. That's your phylogenetic tree that you'll need for your analysis. Because it's closed reference, it's the same, but that's also another perk of closed reference is you don't need to make a phylogenetic tree. So now we want to actually start doing some analyses. And so what I want to do real quick is just I want to make a new directory and copy my important files into it. So go ahead and run the script make directory workshop analysis, and then run the next three. So a CP stands for copy, so you're going to copy your mapping file into your workshop analysis folder. Then you'll go inside your closed reference and take out your OTU table, copy that into the workshop analysis. And then you'll do the same thing. You'll go into your closed reference file, take out your 97 OTU tree folder, and put that in workshop analysis. If yours didn't work like mine did, you can take off these hashtags, and these will save you. So that when you go into your workshop analysis by changing the directory, you should see the tree, the OTU table, and the mapping file. Why doesn't that? Okay, so is there anyone who is not in the workshop analysis folder, or someone who doesn't have the tree, the mapping file, and the OTU table? Because these are important for the next steps.
I'm still waiting for the mic.
You're still close referencing?
But go ahead.
Oh, but you don't have to. If you go up, so see where it says only use this in case you were unable to make the OTU table? So go ahead and do what I did. So leave the exclamation point, but take off the little pound sign. Do that for that one and this one. And you'll also need to do that for the mapping.
Command though, you won't be able to execute a different command until.
But you can stop something running by clicking the giant square here, and then do those other things. And when Damien's up here, he's going to talk about ways of how certain programs filter out singletons, which is a problem in sequencing because you can overestimate your diversity. I take a very simple approach where I just filter out any OTUs that aren't found in at least 3 of my samples. Though I feel like this is, since QIIME 2 is coming out and using data 2, I feel like this is going to be unnecessary soon, and we'll be using these more predictive algorithms. But for the case of this workshop, we're just going to run the script, filter OTUs from OTU table. It's going to take our OTU table, and I just put some O flag as the output. Doesn't really matter the name, it means something to me. And then the S flag has 3, so it knows to remove any OTUs not found in at least 3 of your samples. So then we can go into beta diversity. So this is comparing two populations. So you want to see the differences between your 2 groups. So the first thing we're going to do is run this beta diversity through plot script. It requires an input, so I put in that newly filtered OTU table. My M flag has my mapping file, so it knows what the data means. It wants you to put some sort of output, so I put the O flag beta diversity through plots. So the file that it creates is going to be called beta diversity through plots. And then it also requires the T flag for your tree, just so you can do phylogenetic analyses.
Can you go over like what you just said, but like one more time?
Yeah, of course. Do you mean like the construct of the script, like how the script is broken down, or like what it does?
What you need for beta diversity.
Oh, what I mean. Okay, so we're going to be, it's probably best to go with an example. So what we're going to be doing with this data set is we have fecal samples from knockout mice and we have fecal samples from wild type mice, and we want to see, do their populations differ? What beta diversity does, and this will probably make more sense in a minute, is it calculates distances between samples as a by using certain distance metrics to show whether communities are similar or if they're different. And so that's how we're going to see if the wild type communities are different from the knockout communities or if they're the same.
Ethan, do you want this? Hopefully.
So within this command, I was wondering where you specified which two conditions were being compared.
Oh, great question, but that's not this script. So this script is going to, actually, once it's done, you'll see what it is, but that is important later, and I'll definitely show you. So you guys should be done with the beta diversity through plots. So if you run this iframe that's right under the unweighted unifrac, I didn't tell you guys in the beta diversity through plots thing, but the two most common distance metrics used to calculate distances in microbial ecology data is unweighted and weighted unifrac. They're really great because they maintain the phylogenetic relationships of organisms. So it's just a distance metric that's calculating how different communities are. And so we can, using this iframe a little bit, you guys should see this PCOA plot. Does everybody see the PCOA plot? I'm having a little trouble just because of the sizes, but if you move over here, each of these dots are my samples, right? And you can move this around to see things from different angles. But what's important is to go to the colors and then go under barcode sequence, and then you can change the colors based on any of your columns in your mapping file. And so the only one that was really important to me was genotype. So I want to see, are the communities separating by genotype? And it seems like they are using this unweighted unifrac distance metric, which is kind of interesting. You can zoom in and zoom out, like I seem to keep on doing without whatever. But that's one way you can quickly visualize. But we want to, well, okay. So the next iframe is going to show you the weighted. If we do the same thing, go to colors, change it to genotype, it looks like even on this distance metric that maybe the two populations do cluster separately. And also, if you look at the red dots, they seem to be clustering closer together than the blue dots. And this is something that's ecologically relevant because if you're comparing two communities and you see that one is more similar within the group than the other one, that could infer that there's some sort of control. So weighted, sorry, I should start with unweighted. Unweighted just looks at your populations and says, is this organism here or not? And so it's a very binary approach. So it doesn't take abundance into consideration at all. But if you're doing weighted unifrac, you're looking at the distances, the differences in populations, but you're taking into consideration the abundances. And so with these different distance metrics, you can answer different questions depending on what you're looking for. I use unweighted when I'm looking at more rare populations because it favors lower abundant species because it is just putting everything as binary, whereas weighted weighs the more heavily abundant organisms. So it depends on your question, but it's the binary versus abundance approach. Jackie?
With weighted, would you still see rare populations threshold of when you
Microphone, please.
see it? Oh, sorry.
Don't break it.
Can't be trusted. With weighted, is there a threshold of where you won't see something there if it is in fact there?
That's interesting. I don't think there is a threshold. It depends on your sample because if you have lots of organisms that are largely abundant, you really won't see your rare guys. It's very heavily skewed by your more largely abundant organisms. However, I don't know if you can see this. Do you see how the axes are PC1, PC2, PC3?
Uh-huh.
So those are the principal coordinates. Because there's 8 samples, this real data set, if you weren't looking at it on the screen, is actually a 7-dimensional object because you're calculating the distances between each sample. So it's n-1, but in order to visualize it, because we're humans, you have to ordinate it into a three-dimensional structure. And so PC1 is taking into consideration 41% of the variability, what it can do in order to ordinate. So I wonder if you might change, like if you were to graph different ones, then maybe the rarer species might be able to come up better, but I've never, yeah. Ethan.
So I was wondering if this process of beta diversity is limited to only comparing two different conditions, or if you can do multivariate analysis.
So I want to correct you because this is multivariate analysis already, because each OTU is a variant. But I see what you're saying. You're talking about like 3 different groups. And you can do that. You can have 3 different groups on here. The thing that I run into, and maybe like a real statistician can help me, is I'll show you guys how to do this in a minute, where you actually quantify like whether these are 2 separate groups. And I only know how to do that with 2 samples. So I would take Andrew later and like pick his brain. Okay, so let's actually quantify some of these things. So if you remember what I was saying, it looked like these red dots seem to cluster more tight-knit than these blue ones. And so in order to see if that's a real thing, I can first run this first script, which is beta diversity, and then with the S flag. And the S flag just tells you what possible distance metrics you can use. And I talked about the weighted and the unweighted unifrac, but there's a lot of others, and it just depends on what question you're trying to ask. But like I said, we're using unweighted and weighted unifrac, so go ahead and run this beta diversity.py function that has the input of the OTU table that's been filtered. You'll put some O flags, so I just put beta diversity, and then you put your tree. If the default weren't unweighted and weighted unifrac, I would have to put in another flag and tell it which indices to use, or which metrics to use, but I didn't have to. You can change that. And so once that's done, we can actually look at the distance box plots by making the distance box plot. So go ahead and run the make distance box plot.py. You have your M flag, which is your mapping file. You have your output distance box plots. I named it unweighted because we'll also be doing weighted. And then your D flag, you're going into your beta diversity file, and you're pulling out the unweighted unifrac distance table that you just made. And then if you look, you'll see that your F flag is genotype. And so that can be anything that's in your mapping file, any column. So so my column of interest was the genotype. And then if you put your N flag, you can do nonparametric analyses by telling it how many permutations to do. There's a lot of stuff at the end: suppress all within, suppress all between, suppress individual between. You want to remove these because you're going to be, it automatically makes a bunch of different comparisons, but the more comparisons that you do, you have to correct. And so how the script is just now, we're only making one comparison, so we don't need to correct for multiple comparisons. And so if you run this iframe script right there with the unweighted, we have these two box plots. And so here on the y-axis, we have distance. And so the higher they are on the y-axis, the more dissimilar your communities are within your single group. So here we have the wild type, and we have the knockout. And so looking at this, it looks like all of the wild type communities of my 4 wild type mice, their microbiomes are about as similar to each other as the knockout communities are to each other. So there doesn't seem to be any difference there, but this will probably make more sense. Well, actually, first, let's read the statistics by running the cat distance box plot unweighted genotype stats. You can look at that, and you can see that the p-value, if you're doing non-parametric, is 0.39. So there is no difference, and we could tell that. Looking at weighted, though, go ahead and make the distance box plot with the weighted table. So that should be the only thing different. And then once that's done, do the iframe, and then you should get something like this. And so this is the weighted, right? Yeah. So when you do the weighted analysis, you can see we are actually getting something different. So this knockout, all of these guys, all of the knockout mice microbiomes are more similar to each other than the wild types are to each other, it appears. Now, I haven't done the stats yet, or I haven't looked at them yet, but you can see that just by looking at it like this. And then if we keep going and run the cat distance box plots, look down here, the non-parametric p-value is 0.005. And so we can say that the knockout communities are statistically significantly more similar than the wild type communities. And so this is something that's ecologically relevant. So, but more important, and what most people want to do, is they want to see if those two clusters are actually separate. Are they two different communities, or are they the same? So we can run the script compare categories. The method is Permanova. So Permanova is a type of analysis of variance. It's a MANOVA because it's multivariate data, and it uses permutations to gain significance. So that's why it's called a Permanova. We're inputting the unweighted distance table, which we pulled, wait, sorry, I lost where I am. There we go, which we pulled out of the beta diversity folder. We're putting in the mapping file. We're putting in the category that we're interested in looking, so the difference in the genotype, and then it just wants me to put some output. And then once that's done, we can print the compare categories, and we'll see that the p-value is 0.0259. So it appears that on this unweighted metric that these are two different communities. And so if you guys remember first looking at those beta diversity plots where it seemed like the two were clustering separately, well, it turns out they are statistically significantly clustering separately. And we can do the same thing for the weighted Permanova, so that's just the other distance metric. And we got 0.09, so it's close, but not quite. And so you can see that with these different distance metrics, we are getting different results. And so it's really important to know what distance metric to use to answer the question that you actually want to answer. Namrata? Ethan's taking it.
A little wide.
I don't know whether it's just me, but the same file gives me a different p-value than it gives you.
It's just because it's a permutation and it's probably based on a random number generator that's different. So it won't give you the exact, it should be close enough.
It should be close, but not
it's
exact.
not exact, yes. Unless we actually set this, you can do that sometimes where you set everything to start from the same random number generator, then you would get the exact same values. But for a permutation test where you're doing these, you probably will not get exactly the same number. It won't be 2.5999, it'll be like 2.63 or 2.48 or something like that.
All right, so
it's because when you're doing permutations, you theoretically, given the number of samples you have, you technically can have an infinite number of permutations, so you randomly select some of them. So if your machine is, each of you might be having a different starting point for your random numbers, and therefore your p-values won't be exactly the same unless you force it to do that on the code.
Just a quick question in terms of like when you're actually like analyzing and potentially using this data, if it's off, because mine's off by like 0.02, like is that? Like,
yeah, so it just varies on like how many permutations you run. So because these are probably not, you may want to run 10,000 or like, so that'll kind of make, reduce the differences a little bit. If you run 1 million, you'll.
So you have to like statistically determine what will be.
Well, it's not determining it statistically. It just depends that you just don't want to be at such a small number of permutations that you are going to be, I guess depending on what your level of significance you want to look at. Do you want it to be at the second decimal place or the third decimal place? Okay. So that would depend. That would define the number of permutations you would work on.
Thank you.
all right, so let's go into differential abundance. So we want to actually see if there's a difference in the abundance of certain organisms between these two samples. If you were to go straight into the differential abundance equation, it would tell you the differential abundance of each specific OTU. But for my research, it's a little bit more interesting to look at how the different classes, orders, families, how they fluctuate. And so the first thing I do is I run the summarize taxa script, which requires the input as being the filtered OTU table. You have to put some kind of output. The suppressed classic table output is just, it generates a text file that is unnecessary, so I get rid of it. But you have to put the A flag at the end, because if you don't, it'll put the relative abundance, but you need the absolute count, and you need the absolute count for the differential abundance equation later. So just make sure you have your A flag on there. So now we've generated this file called summarize taxa, which has summarized all of the all of the OTUs at different taxonomic levels. And so we need to get inside that folder. But first, just to make our lives easier, we're going to copy the mapping folder into the summarized taxa folder. And then we can change the directory into the summarized taxa folder, look inside, and we should see the mapping file, and then we should see these 5 new OTU tables. The L2 means phylum, 3 is order, class order, all of that kind of fun stuff. It would probably also help if I showed what it looked like. Oh, never mind. Okay. Anyway, I took this OTU table, and you know what? It will make a lot more sense once we run this differential abundance script. So this is where you actually tell it which categories to look at. So put in the script, differential abundance. You put in the input, which is the OTU table. You're using the DESeq2 algorithm. There are 2 options when you use the script. I like this one because it's more sensitive to smaller population sizes, but it's just a way to estimate the differential abundance. Your mapping file, again, the C flag is your column of interest, so I put genotype. And I know that the 2 variables that are under genotype are wild type or knockout. So under the X flag, I put wild type. Under the Y flag, I put knockout. You can put any 2 things of interest that you're looking at as long as they're under the genotype column. And then my output, I just put differential abundance L2 to separate it from the others. And so once that's done, you can run the cat differential abundance L2 script. And I apologize for the formatting, but we can see that the first guy is proteobacteria. We can see that the base mean is 4,500. Most of the stuff in the middle doesn't matter, except for this number is the log4 change, so it's almost 1. But what you want to look at is the p-adjusted value, which is this guy right here. And so obviously that's a lot higher than 0.05. So looking at this, we can say that there's no differential abundance between the knockout and wild type on the phylum level. But if you go ahead and run the next differential abundance script, this one, everything is the same, except I'm using the L3 file, so we're looking at class. Everything else is the same except the output too. And then once that's done, you print the differential abundance L3 folder and look again. So the first organism is, it's a proteobacteria, but it's class beta proteobacteria. And going towards the end of that, you can see the p-adjusted value is very, very tiny. And so we can see that which direction does the log4 change? So there's 8-fold more beta proteobacteria in the wild type compared to the knockout. And you can do this for any of your taxonomic levels. And there's actually probably more. So delta proteobacteria is also significant. But it's ordered in by smallest p-adjusted value to largest, so you only have to read the first couple to see where your data is actually interesting. The last couple things I want to do, we need to get, so right now we're in the summarized taxa folder. We need to get out, so change your directory with the 2 dots, and that will get you back into the workshop analysis. And then we can talk about alpha diversity. First thing I want to do, though, is I want to look at my filtered OTU table. So I'm just going to run this biome summarized table script. You can see that there's 8 samples. We knew that. Number of observations, so there's 290 OTUs identified. But I use this to look at how many reads are in each sample. So I know that of these 8 samples, the ones with the lowest number of reads is KD8, which only has 3 more than 1,700. So I care about this because I'm about to rarefy my data. And the reason why I rarefy my data is because, as you can see, these different samples have different sequencing depth. And if I were to count the number of unique OTUs that were found in one sample and compare that to another, that will be largely skewed by the sequencing depth. So you can imagine that KD4 with 70,000 reads is probably more likely to have more unique OTUs than KD8 just based on probability. So what I'm going to do is I'm going to rarefy, which subsamples all of these different samples to a specific sequencing depth. So I'm going to set that sequencing depth to just a little lower than 1,700, which will preserve my file that has the least number of sequencing reads. And so that's the single rarefaction function. You put in your input, your OTU table. You have some sort of output, which I call OTU filtered, rarefied. And then your D flag tells it how many, what your subsampling depth is going to be. If you want to know more about rarefying, it seems to be a slightly controversial issue, and I would recommend Susan Holmes' waste map paper. Sorry, Ethan, this was literally like the first time I looked up in a while. How's it going?
I was just wondering, how does it choose which reads to exclude in that process?
It's random. So you could, and that's a really important point, because in the single rarefaction, you only rarefy it once. And so whatever you get, you get. You can do the multiple rarefaction script, which will do it as many times as you want. So if I tell it to do it 10 times, it'll rarefy it 10 times. And then I can do certain types of analyses with that. But it is random. There is an important distinction. If you want to rarefy with or without replacement, and I think most people do without replacement because that seems to be more biologically relevant. But if you're subsampling and you're letting, if you sample one guy and you let him come back, that's not, it's a little weird. So I like if you're subsampling and just like not letting him return. And that seems to be the default. Yeah, okay. So that was the single rarefaction. So then we can do actual alpha diversity stuff. So go ahead and run this alpha diversity script. You put in your OTU table, the rarefied one, and then you'll see with this M flag, you can put different diversity indices. So I like to look at the observed OTU, so that's literally how many OTUs were found in your sample, the Shannon Diversity Index, Chow-1, Simpson-y. They all have important ecological relevance. You put in your T flag as your tree, and then you put in some output flag. And then, once that's done, we can look at our alpha diversity, and it will give us these numbers, which don't mean a lot now, but you can plug this into GraphPad, and you can look at if there's differences in OTUs, observed OTUs, if there's trends in Shannon diversity, all that kind of fun stuff. But go ahead and run this alpha rarefaction script, because it takes 20 minutes, and we probably won't even have time to go through it, but we can at least talk through it. So as Chris was saying earlier, this is great. You can see, you can rarefy your data and you can compare between samples, but a great way of visualizing your data is to make these rarefaction curves. And so if you remember on the x-axis, it had all the different subsampling, and then on the y-axis, it had how many unique OTUs. And so you can imagine the more you subsample, the more unique OTUs you'll find. And so you can see how your data's distributed. So honestly, like not gonna have time to finish this, but when you're done with it, you can do the iframe script at the bottom. It will tell you, it'll give you like 3 different diversity metrics you can use, and then it'll tell you like which column do you wanna organize your data by, and then you can see how the rarefaction curves show. But other than that, I think I'm all done, and I think Damien really wants to talk about data too at some point. Yeah? Okay, great. Does anyone have any questions before I go? Great, thanks guys.